أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والمرسلات عرفا فالعاصفات عصفا والناشرات نشرا فالفارقات فرقا فالملقيات ذكرا عذرا ونذرا إنما توعدون لواقع فإذا النجوم طمست صدق الله العلي العظيم. Lecture number 8 for first year class. And two structures. Normally we think these structures are not much needy structures in the plants and the animals. These are the backbones for the food for the animals, because seed will not grow, the plants will not grow, producers will not produce the food, and carnivores and herbivores will not be able to eat the plants. Second one, second organelle, which is a hydrogen peroxide, lot of alcohol some people are drinking in any format all over the world. Alcohol detoxification, even in our religion, that is not good. Still nature gave one organelle to detoxify the alcohol. Alcohol is a toxic molecule. Ethanol, propanol, butanol, pentanol, hexanol, heptanol, octanol, nonanol. Most important alcohol which is being used, that is ethanol, C2H5OH. That ethanol detoxification take place by peroxisomes. Two organelles I am naming, peroxisome and glyoxysomes. These two organelles are unique organelles. Normally, unlikely, we are studying mitochondria, Golgi complex, endoplasmic reticulum, nucleus, lysosomes, and you was giving importance to those organelles. But when you will study the function of these organelles, you will understand how electron transport chain is taking place in our body. And you will also understand how the photorespiration take place in the plants. A new terminology, if I will say in simple words, plants has two type of respiration. Plant has two type of respiration. One is a respiration, one is a photorespiration. Photorespiration mean in presence of light. Mean at the daytime the plant is doing two type of respiration? No. At the daytime the plant is doing only one type of respiration and one type of photosynthesis. But when photosynthesis is not taking place, photorespiration take place. I will explain this thing in coming lectures also, because that is the topic in your book. But one thing I will tell you, photorespiration is against photosynthesis. Anything which is against photosynthesis, that is synergistic with the respiration, mean oxygen gas will be used and carbon dioxide gas will be released. Now you are understanding the benefit of glyoxysome organelle in the plants. Why it is present? Due to photorespiration. Due to photorespiration. Peroxisome, photorespiration. Hydrogen peroxide, peroxisome. Glyoxysome, to another function of glyoxysome, to change the fats into the starch. No seed can grow if the glucose is present. No even living thing can grow that even the glucose or starch will be present, because starch can undergo respiration directly. The fats molecules cannot go. We have the fats. Our fats, if we will not eat the food, our fats will be burned. That will be changed into the starch by enzymes. But glyoxylate cycle I am talking about in the seed. In some seeds in which lipid stored seeds, all seeds are not lipid stored seeds, in which fatty acids are stored, in which the glycerols are stored, how those seeds will get the energy? One cycle, glyoxylate cycle. The glyoxylate cycle will change the starch, into the now detoxification of alcohol. But I will peroxisome take place by peroxidase enzyme. Two enzymes are used to detoxify alcohol. Two enzymes are used. One enzyme is peroxidase. One enzyme is a peroxidase. This enzyme is used, peroxidase. This enzyme will change alcohol into hydrogen peroxide, which is still harmful. Peroxisome organelle, which is present in the living thing, it is present in the plants as well as in the animals. I am talking about the two extraordinary organelles. One is the glyoxysome. I am talking about this organelle. I am talking about another organelle, peroxisome. These two are extraordinary organelles because they are doing extraordinary works. Already I told you they are detoxifying alcohol. Peroxisome is detoxifying alcohol. Alcohol changed to hydrogen peroxide. Alcohol changed, this is alcohol, goes to hydrogen peroxide, and then it goes to water, then it goes to water, H2O. So by this way this cycle will complete. But another cycle, glyoxylate cycle by glyoxysome in the plants, that is important. I will name one amino acid as well, glycine, but before that I will tell you photorespiration. What is the photorespiration? The respiration which takes place in the plants when photosynthesis is not occurring. Which gases are being exchanged during photosynthesis? Carbon dioxide will be used and oxygen gas will be released in the photosynthesis. Oxygen gas will be in the photosynthesis. So photorespiration takes place when carbon dioxide, which is the raw material for the photosynthesis, that is not available, and carbon dioxide is not available. Normal respiration is not producing much carbon dioxide. You need one another process, which is photorespiration, which will produce also carbon dioxide. As the carbon dioxide become normalized, photosynthesis will start once again, because that carbon dioxide will be available for the plant. That was the reason. Very long cycle of photorespiration in chapter I will discuss, but one thing I want to tell you, glycine, 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 which is amino acid, that glycine formation is small step. Photorespiration is not simple process, it is in the chain form. One reaction, it is in the chain form. One reaction take place, second reaction take place, third reaction take place, and then one reaction is the conversion of glyoxylate into glycine, which is amino acid, that take place in peroxisome. That takes place in the peroxisome. Now we are going to discuss some slides, because that I will come back to topic as well in the detail, but these slides will help in understanding of the topic. Glyoxylate cycle, students, where it is present in glyoxysomes. Glyoxysome, what is the glyoxysome? It is the cell organelle. It is the structure inside the cell, extraordinary structure, present in plants or animals, present in plants. What is the function? In all seeds, present in all seeds, not in the plants, in the seeds of the plants. In all seeds of the plant it is present. In all seeds of the plant it is present. In all seeds, no. Lipid rich seed, it consists of in the lipid rich seed, and they are not found in the lipid poor lipid seed. Lipid is also called fats, mean fatty acid glycerols are present. So glyoxysome is important, because in glyoxysome another cycle is completed, glyoxylate cycle. What is meaning of glyoxylate cycle? Important. Fats changes to starch, starch. Fats changes to starch. Fatty acid glycerol changes to starch. Why there is need to convert the fats into starch? Because starch will not change into glucose. The normal reactant of respiration is glucose, and glucose will not react with oxygen, and respiration reaction will not take place. Carbon dioxide, water will not be produced, energy will not be released, and this respiration reaction, cellular respiration reaction in mitochondria, depending upon this glyoxylate cycle, because this cycle will convert F to S, mean F will change into S. You can understand the fats, because fats cannot take part directly into the respiration. We go to another slide. Glycolic acid oxidase from glycine in peroxidase. I was talking about this phenomena. I was talking about this significant reaction taking place in the peroxisome. Glycolic acid oxidase from glycine. This is enzyme, glycolic acid oxidase. This is enzyme, and it will form the glycine. Glycine, I told you, glycine is an amino acid. You want to see the word also under the glycine is an amino acid. Glycine is an amino acid, and glycine, what the glycine is doing, that what is the advantage of the formation of the glycine in the peroxisome? When you will study photorespiration and you will start from A and you will finish at the Z whole cycle, then you will understand when glycine will not be formed, carbon dioxide will not be released as a result, because the major objective of photorespiration is to produce carbon dioxide. Just told a moment before that photorespiration taking place because respiration is not enough to produce enough carbon dioxide, and carbon dioxide is not being absorbed from outside. You know, carbon dioxide atmosphere supply by stomata, that is not available. So plant in the condition when clouds are there or some other problems are there, carbon dioxide concentration is less, so plant will produce its own carbon dioxide by another respiration, and glycine formation is integral part of photorespiration. Enzyme of the glycine formation take place in which organelle? Peroxisome. And this peroxisome enzyme, which is present in the, when it is present in the plants, what it will do, it will convert the glycolic acid oxidase, it will convert the glycolic acid into glycine by the enzyme glycolic acid oxidase. We go to another slide. The glycolate changed into glycine. That was the point. In the photorespiration inside peroxisomes, the peroxisomes are very important organelles in the plants, because what they are doing, they are converting the glycolate, a reactant. Don't worry about the glycolate name, because when I will teach you Melvin-Calvin cycle in the photosynthesis, then you will understand, because in the photosynthesis what are the reaction? Light reaction, dark reaction, Melvin-Calvin cycle. You know that Melvin-Calvin cycle normally is doing photosynthesis, but in this case it will produce a 2-carbon molecule glycolate, which will undergo a different processes, and one of the processes is that it will enter into a organelle. The name of the organelle is peroxisome, in which the glycine will be formed, and that glycine will be used in the photorespiration in the next step to produce the carbon dioxide. And in chapter we will study photorespiration in detail. In plants seedlings stored fatty acid, the point I was talking about. Now I think easier, because I decrease the size of the picture. Now visibility is there. Thank you very much. So now see, in plants, seedlings stored fatty acid change to starch. Seedling, when the seed is growing, when the seed germinate, little bit some roots and stems, plumule form, stem, root, plumule form, stem. Two structures in the seed, you remember. Plumule form stem and radical forms root. When the seed grow, this is called the seedling. In the seedling, when the seed is growing, the term you will use, the seed is germinating. When the seed is germinating, the seed will consist of, some seeds, or not all seeds, castor bean plant, in which the lipid rich seeds are present. When the lipid rich seeds are present, they are full of the fats. They cannot undergo the metabolism, they cannot undergo the catabolism, they cannot undergo the anabolism. They will not participate in the respiration, because respiration is integral part, either the plants or the animals, any cell is there, respiration, cellular respiration, which takes part in the mitochondria. It will not take place, the things will not remain alive. So reactant of respiration is glucose. Question, sir, here you are forming starch and you are telling the main reactant of the respiration is the glucose. When starch is formed, starch belong to which family? Starch belong to which family? Carbohydrate family. And carbohydrate family, polysaccharide. Remember, polysaccharide, next chapter I have to teach you. Next chapter or I think after enzymes we will study these organic molecules in detail. Polysaccharides, disaccharides, the monosaccharides, the starch is polysaccharides. In short, I will tell you, starch belong to carbohydrate family. Of course it is not glucose. Of course it is not monosaccharide, because glucose is monosaccharide.
but starch is a polysaccharide, but we can convert very easily because they belong to one family. So starch will change into the disaccharide, mean sucrose, and then it will change into glucose, easier one. But when it is in the form of the fats, they are unable to convert into the glucose. So therefore this glyoxylate cycle is very important because it is changing revolutionary organelle. I give you example by considering your body. You are doing exercise. What you was doing? Why you are doing exercise? You was burning your fat. So where the fat is going? Because you was exercising. Just like a vehicle run by petrol, your body run by glucose. So what happened? Fats and glucose, no relationship. So why you was burning fats? So where the fat is burning available, there is no fire in the body and fat was not visible. In abdominal cavity, most of the fats molecules are deposited in the peritoneal cavity because here are some layers, air spaces are there to deposit the fats. When you will not eat much and you are exercising, energy required more, the body decide, the nervous system decide that we are not fulfilling the requirement of the glucose from the starch. So we need to get the glucose from the fats because that is option. But if you have the starch, if you have enough food, even you are exercising, the fats will not burn. So you have to take we have to take control of diet as well. So if the starch molecule, which is being changed to the glucose, starch molecule which is being changed to the glucose, and starch molecule is less available during exercise, our nervous system decide to convert the fats into starch, one type of organic molecule into another type of organic molecule. So we need enzymes. Similarly, plants also use glyoxylate cycle, and specific organelle is being given. That organelle will convert the fats into starch. That is the road, that is the map road to follow, because when starch is formed, it will go into the disaccharide very easily by decomposition, and also it will go into the glucose monosaccharide very easily, and by this way they can participate in the respiration reaction, they can react with oxygen, they can form carbon dioxide and water, and energy will be released, and that energy will be used. The point which I wanted to share more, because I discussed this thing, peroxidase enzyme present in peroxisome, detoxifying alcohol. There are two toxic molecules written in this slide. One is a alcohol, this is also harmful chemical. One is a hydrogen peroxide. They both are dangerous. So the harmful effect will not finish until the water will be formed. So first high level harmful product is the alcohol, number one. Second one is hydrogen peroxide. The third one is the water. So two enzymes are being used by peroxisomes. One enzyme will convert ethanol. I use the name ethanol, formula is C2H5OH. The first is methanol, ethanol, propanol, but ethanol is mostly used in the living things, and ethanol C2H5OH will be converted into hydrogen peroxide H2O2. Now you are understanding why peroxisome is used in this reaction. Main work of peroxisome is to oxidize hydrogen peroxide or to reduce hydrogen peroxide. I will define oxidation reduction very quickly. Addition of oxygen, removal of electron, removal of hydrogen is called oxidation. Very easy, O plus E minus H minus. Reduction, removal of oxygen, addition of electron, addition of hydrogen is called reduction. We discussed in the class. Oxidation reduction collectively called here. These are the centers. Peroxisomes are the center where oxidation reduction reaction take place. I give you one example. H2O2 changes to water. So what you was doing? Formula of water H2O2, formula of hydrogen peroxide H2O2, formula of water H2O. What you did? You remove oxygen from the hydrogen peroxide. Who remove oxygen? Catalase. So catalase was reducing hydrogen peroxide. So catalase was reducing agent. Where it was produced? In the peroxisome. So peroxisome contain the enzymes who can oxidize, who can reduce. the chemical who oxidize other is oxidizing agent, and chemical who reduce other, that is reducing agent. Hydrogen peroxide reduces to water because oxygen atom is removed, so catalase is a reducing agent. Present where? In the peroxisome it is present. In the peroxisome it was present. So we go back to our another slide which I wanted to show you, but before that I will tell you one thing, and this is the, I think, major information, and you will be able to understand what I wanted to say in this lecture. Photorespiration word I have used, photorespiration the word I have used, and I mentioned that that is the extra process takes place in the plants when it takes place when carbon dioxide concentration is not enough to fulfill the requirement of the photosynthesis, and another reaction take place, and both respiration take place. One is a normal respiration, one is a photorespiration. Questions are, without photorespiration the plants can remain alive? Yes. Photorespiration is a temporary processes take place, and with photorespiration all the time do not take place. Some scientists are also thinking why photorespiration occur. So this is the answer, that photorespiration occur to fulfill the requirement of the carbon dioxide. Still the scientists are researching, because the photorespiration will not occur, the plants will remain alive, but photosynthesis abnormality will induce photorespiration. This is the point. Other researches are being going on, and one point they raised, that carbon dioxide concentration is less, so the plant need more carbon dioxide, so plant decide to undergo an abnormal reaction of photorespiration using Melvin-Calvin cycle, which in the next chapters we will study. Catalase enzyme I used, catalase enzyme, and one is a glycine. Glycine is formed in the amino acid by glycolic acid oxidase, you study. And glyoxylate cycle, where it is present? This catalase enzyme was present. This catalase enzyme was present during, during the decomposition of the hydrogen peroxide. I think I have to explain once again. Alcohol is toxic molecule. Alcohol is toxic molecule. Okay, formula of ethanol is C2H5OH. Alcohol changes to hydrogen peroxide by peroxidase enzyme. One reaction, clear? This is one reaction. The hydrogen peroxide is still harmful, and we have to reduce hydrogen peroxide to water. Which enzyme will be used? This enzyme will be used. It will break the hydrogen peroxide H2O2 into water. It is written here, you can see. Okay, we will discuss now glyoxylate cycle. What is the glyoxylate cycle? Glyoxylate cycle is the cycle in which, once again I am telling you, lipid rich seed to convert the fatty acid into the carbohydrates. To convert the fatty acids into the carbohydrates means starch. When starch family is formed, glucose is formed, because enzymes to convert the starch to glucose are present in our body, in the plants, and those glucose will take part in respiration by reacting with oxygen to form carbon dioxide and water in the mitochondria. In much detailed phenomena of electron transport chain and Krebs cycle, and lucky thing is that these all reactions is much detail are included in your syllabus of the first year. Krebs cycle, etc., the light reaction, the dark reaction, the Melvin-Calvin cycle, and photosystem 1, photosystem 2, antenna complex you will study in the next chapters, and hope you will revise this one. Okay, thank you.